Welcome to 1.4, Membrane Structure and Function. What you're going to learn today is the components of the membrane and their function. We're going to learn about transport proteins and how they work, what makes one active and the other one passive. We're also going to learn about the requirements of the movement of water through the membrane. Come along as we explore membrane structure and function.
On the structure and cell size, and that's membrane structure. So let's revisit our friend the phospholipid. Remember, the phospholipid is a lipid because it has fatty acid tails and it has a glycerol, but it also has a hydrophilic head. Because it has both hydrophobic fatty acid tails and a phosphate that is hydrophilic, we call this an amphipathic molecule. And what this means is it's a molecule that can do both. Part of it likes water, part of it does not like water. And so the head with the phosphate is going to want to associate with water, whereas the fatty acid tails want to be as far away from water as possible. This allows for the bilayer formation, or the foundation of the membrane structure. And you can see in the diagram we have a single layer sphere, a bilayer sphere. That would be very similar to a vesicle. And then your bilayer sheet, which is what the membrane would be. So there's your hydrophilic head, and there's your two hydrophobic tails. Your fatty acid tails can be both unsaturated or saturated. Remember, that means if there is a double bond in the carbon backbone, then it's unsaturated because you do not have the maximum number of hydrogens. And you are bent because it's a cis formation. So what does this have to do with membrane structure? Well, the fatty acid tails play an important role in membrane fluidity. So there is movement of the phospholipids, which allow the proteins and other molecules embedded in the membrane to move around. So there is lots of lateral movement. However, you do not want your membranes to have too much fluidity, or the weak interactive molecular forces, the weak intermolecular forces, are going to break apart. Okay, so with an increase in temperature comes an increase in fluidity. Okay, and with a decrease in temperature, you get a decrease in fluidity. You never want your transport, you never want your membrane to stop moving, or what happens is you're no longer transporting substances. The cells aren't getting what they need, and so they're not going to be able to function for very long without this movement. Okay, so if you need to increase the fluidity, like cold weather, cold adapted organisms, then you're going to have lots of unsaturated fatty acid tails, okay, because that bent fatty acid tail is going to act as a spacer. It's going to push the molecules far apart, preventing them from stacking, well, not far apart, but prevent them from stacking close to each other and solidifying. Saturated fatty acids, on the other hand, your straight chain ones, are going to decrease your fluidity. So what this is going to do is, in your warm adapted environments, you're going to have more saturated fatty acids to decrease the fluidity. Remember, the higher the temperature, the more fluid you have. Now for plants have that cell wall. Okay, so they just are going to have the unsaturated and saturated fatty acids to maintain their fluidity. Animals, on the other hand, don't have a cell wall, so if their membrane cohesiveness breaks down, then there's problems, major problems. Okay, the cells are no longer functional, your organelles and cytosol are flowing out. So cholesterols play a role in maintaining membrane fluidity, and these are found in animal cells only. And when we look at membrane structure, we'll see that the cholesterols are going to be embedded between some of the phospholipids. And what this does is it acts as a fluidity buffer. It's going to act as a spacer. It's going to hang on to those phospholipids to slow them down or space them out, depending upon what is needed, if we need it to be more fluid or less fluid. So let's look at membrane structure. You have a sea of phospholipids as your canvas. This is with your proteins and various molecules embedded in the bilayer. Okay, membrane is described as fluid because the molecules do move around. There is some anchorage with the cytoskeleton, but not a whole lot to keep the membrane rigid. It still moves, it still flows. Mosaic because you have different membrane components on the outside of the cell and on the inside of the cell. Some proteins are transmembrane. They go all the way through. They span the membrane. Other proteins are peripheral. They're going to be only on one side. So membrane structure is fluid. The components can move around, and other molecules are going to be embedded throughout the membrane. This allows for the membrane to carry out various functions to allow for selective permeability, to allow only certain materials in and other materials out. So we have some components of the cell membrane, lots and lots of proteins, each with lots of different functions, different jobs. Some of them are going to tell the cell what to do. Some of them are going to transport substances into and out of the cell. Some of them are going to anchor the cytoskeleton. Some of them, like the cholesterol, are going to act as fluidity spacers. Glycolipids and glycoproteins are going to have oligosaccharides or short polysaccharide chains that are added as ID markers. This is how your immune system identifies your cells as yours. Your markers, your set of markers, are going to be different from somebody else's. Okay, this is how, when we get to genetics, we'll talk about blood type A and blood type B, but that particular gene codes for two different markers, and what we're talking about is these glycoproteins with these carbohydrate chains. Very important molecules of the cell because they act as identifiers. They're going to tell your immune system if something fishy is going on in the cell, like a virus reproducing. So these are very important, play a very important role in recognition of the cell. So there are several different proteins besides cellular recognition. We have transport proteins that transport various substances. You have signal reception proteins, attachment proteins for support, of course, cell recognition. Enzymes are going to be embedded in the membranes, and cell junctions. And when I'm talking about cell membranes, I'm not just talking about the outermost membrane. That's what we talk about a lot, but we're also talking about all the membranes of the nuclear envelope. We're talking about the membranes of the ER. We're talking about membranes of the Golgi apparatus. All of these are going to have proteins and transport substances and signal receptor proteins and enzymes embedded in these membranes to do and help with those organelle functions. So the membrane can only be the cell can only be as big as it can transport in wastes and bring in the amount of nutrients it needs. The bigger it is, the more waste it produces, the more nutrients it needs. So there are restrictions to cell sizes. We don't have, the bigger the organism, we don't get bigger cells. The cells stay the same size. Well, it all has to do with transporting substances efficiently into and out of the cell. Okay, the bigger the cell gets, you have a smaller surface area to volume ratio. So you don't have a whole lot of transport mechanisms to get everything the cell needs into that large volume. So it's inefficient to move substances across the membrane the larger the cell gets. However, the bigger the surface area to volume ratio, the more surface area you have with a lower volume, the more efficient the cell to exchange the material with the environment. This is why in cells and in tissues and organs we see folds. We're going to see a lot of folding patterns. Okay, we saw it with the microvilli. We see it with the ER. Those folding patterns that we see, its main reason is to increase the surface area so we can have more exchange, bring in more nutrients, transport more substances, and increase the efficiency of the cell and the cell's processes. And this concludes the first part of membrane structure and function.